Mosquitoes like these ones are very bad news for us because they actually transmit many infectious diseases that kill people. And on top of the list of dangerous infectious diseases is definitely malaria. And we all know that malaria kills, but maybe not everyone knows that malaria kills a lot of people. And in fact, since the beginning of this century, in just over 10 years, malaria has already killed 10 million people. And that's almost like 1 million people every year. And on top of that, malaria also infects more than 200 million people every year, causing huge social and economic problems to the countries where malaria is transmitted. But I believe, and today I'm going to talk about the fact that I believe that this can be stopped. We can stop this, so malaria can be eradicated. It's not going to be easy, it's going to take time, however, it's feasible. And if we look at the map of the world, according to the number of malaria deaths, we see a very different picture that we're used to. We see that there is a lot of Africa on this map, and that's because 90% of malaria deaths happen in Africa and mainly affect young children before they can develop immunity. So it's pretty fair to say that the mosquitoes that transmit malaria in Africa are the most deadly organism that we have in the world. And how do these mosquitoes transmit malaria? When a female mosquito bites a person that is infected with the parasite, then she will take the blood and the parasite, and next time she bites another person, then she will inject the parasite into the bloodstream and will spread the disease. So if we stop the mosquito, we stop malaria. And this might sound very difficult. However, the good news is that we don't have to stop every mosquito that we have in this world. In this world, we have thousands of mosquito species, but only a small number of these mosquitoes can transmit malaria. About 10 to 20 species are good at malaria transmissions. So it's enough to stop this. And we already have good tools to do that. In particular, we have insecticides that we can use on mosquito nets that we use to protect people while they are asleep. And so that when mosquito comes and try to bite mosquitoes that are, try to bite people that are asleep, then they get killed. Or we can spray these insecticides inside the houses, on the walls. And so in that way, we can kill mosquitoes that are resting. And these techniques, these methods are very good and they're working. However, by themselves, they cannot stop a disease that kills almost 1 million people every year. And moreover, there is a problem. It's that mosquitoes are becoming more and more resistant to the action of these insecticides, which means they're not killed anymore. And in some cases, they also change their behavior. And rather than biting inside the houses and during the night, they start biting outdoors and during the day, so that we cannot reach them and insecticides cannot kill them. So what can we do about this? This is our idea. Our idea is, why don't we use sex to control mosquitoes and stop them from transmitting malaria? So if we actually make mosquitoes sterile rather than killing them, then we can interfere with reproduction, and then eventually there'll be fewer mosquitoes out there that can transmit malaria. And of course, stopping mosquitoes from reproducing is not easy. It's quite tricky. However, the good news is that female mosquitoes have sex only once in their lifetime. So they have sex once, and then that's it. They stop. They won't have sex anymore for the rest of their lives. So if we can interfere with these single sexual events, then we can really induce sterility for the female lifetime. So this is our idea. Why don't we actually generate thousands and thousands of males that are sterile, or even millions of sterile males, and we release them in the field. And then these males will do the job for us. They will go and find those females that are so difficult for us to find, and then we'll have... Exactly. And they'll find those 17% of females, and they'll have sex with them. And however, there will be no progeny coming out of those matings because the males are sterile. And if we do this over and over again, eventually the population will crash and there'll be no more malaria transmission. And it might sound like a bit of a horror movie for you to imagine millions of males released in the field. However, actually, these males are pretty harmless. They don't bite, they don't transmit disease. The only thing they have in mind is sex, and they'll go for it. And so, at the end of the day, we can use mosquitoes to eradicate mosquitoes. And this is not a new concept at all. Actually, people in the 70s, like last centuries, they already started thinking along these lines. However, back then, the only way that they had to sterilize males was through the use of chemosterilants or the use of high dosage of gamma radiations. That yes, they would achieve sterility. However, they would also affect the general biology of those males, so that when those males were released, they were pretty useless and they wouldn't be fit enough to compete with males born and bred in the field, and so they would not be able to have sex. But now we can be much more subtle about this, and we have the right genetic and molecular technologies to actually induce sterility without affecting other aspects of mosquito biology. And that's what we do in the lab. We study the mosquito DNA to identify what are the factors and mechanisms that are important for mosquito fertility, so that then we can interfere with these factors specifically and induce sterility without affecting other aspects of mosquito biology. And we have already been able to do this in our lab. So what we've done, we have generated males that have no sperm. And here you can see sperm from a mosquito male. And we make it fluorescent, green fluorescent in this case, so that it looks beautiful, and also we can visualize it, and then we can interfere with the formation of sperm. And so when we develop spermless males, we can see the absence of sperm, and we can selectively take those males and use them for mating with females. And while normal females, after mating, they develop hundreds of eggs, in this case, they'll produce 0 progeny. So there'll be no progeny coming out of those males. So this is really good news. However, there is a problem, is that this process is very inefficient. It's highly inefficient. And the way we do it in the lab, we take females, we feed them on blood so they develop eggs. Then they lay the eggs, and we take them one by one. We align them onto glass slides, like these ones. And then we put these glass slides onto a microscope, and we start injecting with a joystick these mosquitoes with needles that are filled with an inhibitor that prevents sperm formation. So we do these injections in these embryos one by one. And this is a highly inefficient process that also kills a lot of the embryos. So it's quite traumatic for the embryos. And so if we start in a day, we can inject like 1,000 embryos. And at the end of that day, we'll have 20, 30 males that have no sperm. So this is good for our studies, but it's definitely not good for a field application. So how can we scale this up and go from these 20, 30 mosquitoes that we can produce at present to the 20,000, 30,000, even a million of males, spermless males that we need to release in the field? So we have different opportunities, different possibilities to do this, and that's what we are working on in the lab. So we could use, for instance, transgenic technologies. We could genetically modify these males and incorporate the inhibitors in their DNA, so that then all the males would express these inhibitors, and we could suppress this in the lab and then have the inhibitor activated in the field when we release the males. However, this is something that is work in process, but in progress. However, there are some issues before this can be implemented. In particular, there are some regulatory issues concerning the release of genetically modified organisms that maybe one day they will be sorted out, but they would limit this approach at present.
Didn't Brazil do this for dengue?
Yeah. Yes, these are something that, yes, but it's used in America, in Northern America, in Brazil, but it's not been never used in. It's never been used in Africa. Yeah. But the second issue that is maybe more relevant is the fact that we will need to modify the genome of every single species that we want to target. And so, although this is feasible, it's a little bit limiting. It would be a limiting step. So what if we instead scaled up what we already have now, and so scaled up the use of these sperm-forming inhibitors that we have already available? And so, for instance, what if we had automated machines, like robotic machines, that could align these embryos for us and then inject them automatically with sperm-forming inhibitors? Or even better, what if we could inject these inhibitors into the mothers? So if we could actually inject them into females and then have them incorporated into the embryos while they are developing, so that then once they are laid, every embryo would contain already the inhibitors without the need for us to inject them one by one. This would be a much more efficient way. And then the same system, so the same inhibitors could be used over and over again to target all mosquito species that transmit malaria in Africa, and maybe not just mosquito transmitting malaria, but also other infectious diseases. So we could use the same system for many different insects. And then people say, yeah, but what about the environment? What about the food chain? What about the ecology of this? Are mosquitoes good at something? And these are valid concerns. However, we should consider the fact that there are thousands of mosquito species, while this would be a very highly selective way to target only those that transmit malaria. And unlike insecticides that kill every insect, this instead would only work with the mosquitoes that we want to target. And in terms of the logistics of an operation like this, there's already a lot of expertise out there concerning releases of sterile insects that are a problem for agriculture. We have, as we speak, there are millions of flies that are agricultural pests like fruit flies, melon flies, potato weevils, screwworms, you name it, that are being released, sterile insects that are being released all over the world. And so there is a lot of expertise in terms of packaging, distributing, and releasing these insects. And so with a system of distribution that already exists, and we could translate this system into mosquito work, and with also in integrating these methods for mosquito control with already existing use of insecticides, and of course also with the use of drugs to kill the parasites in people, and together with a good system for monitoring the progress of the program, and also with the commitment from national governments and international agencies, then we could really have a recipe to give a fatal blow to malaria and finally see the end of the disease. And thank you for your attention.